In some ancient drawings, not only can the trace of a master be hidden, but also the biological traces of the one who touched him. An international group of scientists has begun to explore the possibility of extracting human DNA from Renaissance works of art. At the center of this research is a small red chalk sketch attributed to Leonardo da Vinci and known as Holy Child. If you like the content we are sharing, do not forget to subscribe and give like. This study, even in preliminary phase, holds that this work could contain genetic traces compatible with the paternal lineage of the famous Italian artist. The research, published in January 2026, combines state-of-the-art genetic sequencing techniques with non-invasive sampling methods. Authors call this technique biological signatures of history. Among the most relevant findings is the detection of markers of the Y chromosome in the drawing and in a letter of the 15th century. Both share genetic elements that researchers associate with the same ancestral line, possibly the Da Vinci family. This innovative approach seeks to trace the past without damaging valuable heritage objects. One of the biggest challenges in working with these objects is the need to keep them intact. Traditional techniques to obtain DNA are unthinkable in materials as fragile as a Renaissance drawing. Therefore, a sampling protocol based on the use of double hyssop was developed. This protocol allows to collect traces of genetic material without damaging the surface of the works. It was applied to a selection of works, including the Holy Child drawing and several letters from Francesco di Ser Giovanni da Vinci, relative of Leonardo. After sampling, the researchers performed a complete metagenome sequencing, allowing them to detect mixtures of bacterial, fungal, plant, animal, and human genetic material, establishing unique biological profiles for each object. Holy Child was the only object analyzed that showed detectable human DNA. In 3 different swabs taken from the drawing, specific markers of the Y chromosome were identified, corresponding to different branches of the E1b1b haplogroup. The letter written by Francesco showed coincidences with the E1b1 branch, reinforcing the possibility of a familiar link between the drawing and the artist. However, the authors themselves insist that these results should be interpreted with caution. One of the highlights of the study is its conservative approach to the interpretation of the results. Despite the emotion that can be generated by the possibility of having found Leonardo's DNA, the DNA present is extremely fragmented and contaminated. A Holy Child drawing is the visual heart of the study, but its authorship remains widely questioned. This ambiguity has direct consequences for the interpretation of the results of the genetic study. This study represents a milestone in the integration of genetics with the history of art. Although it poses limitations, it marks a clear roadmap for future research on the genetic profile of historical figures.